My name is Janae Heiss, and this video is on the topic of Mendelian genetics. I am doing this video for my master's degree in Applied Ecology and Conservation Biology at Frostburg State University. My thesis for this degree is on cannabis cultivar genotype and chemotype, which is basically how different types of cannabis look in their genetic composition and in their chemical composition. This concept of Mendelian genetics pertains to my thesis because Mendelian genetics is the foundation of genetics, and my thesis involves microsatellite genetics. So this video, per Dr. Putoff's request, is 60 minutes long and does not use slides or notes. In this video, we will be covering the background for Try again. In this video, we will be covering some background for Gregor Mendel, sort of his life history, background of his experiment with Mendelian genetics, some background for what he was facing when he was doing this as an experiment. So the dominant theory of the time, which would be blending, and how he proposed a new one involving elementin. Then we're going to get into what exactly Mendels experiment was and some definitions of genetics that are important to know. We're going to get into monohybrid crosses, dihybrid crosses, and Punnett squares. And then we're going to look at the rules of inheritance and the 3 principles of inheritance. We'll also get into pedigrees. Then, after we've covered all of the Mendelian genetics, we're going to get into non-Mendelian genetics. Non-Mendelian genetics includes things like sex-linked genes, incomplete dominance, codominance, multiple alleles, so when you have multiple alleles for a gene, pleiotropy, polygenic inheritance, epistasis, and that's the main ones that are not Mendelian genetics. We will also get into environmental considerations. So, environmental considerations or phenotypes. And lastly, we will get into the current research that is occurring on Mendelian genetics, because this is a concept that is still important for genetic research today. Mendelian genetics. There once was a man named Gregor Johann Mendel. He was an Austrian monk with a fascination for peas and heredity. He was a gardener, an astronomer, theologian, physicist, and beekeeper. Due to his famous experiments, he is known as the father of modern genetics. The way in which traits are passed from one generation to the next, and sometimes skip generations, was first explained by Gregor Mendel. Mendel developed 3 principles of inheritance that describe the transmission of genetic traits before anyone even knew that genes existed. Mendel's insight greatly expanded the understanding of genetic inheritance, and it led to the development of new experimental methods. Let's talk about Gregor Mendel. Who exactly was he? Gregor Mendel was born July 20th, 1822, in the village of Hensendorf bei Odrau, in the district of Moravia. This was part of the Austrian Empire, and is now the eastern area of the Czech Republic. He was baptized as Johann Mendel on the same day. He did farm and field work from his childhood until early adulthood. He went to the Piarist school because of its free tuition growing up. And then he went to the Philosophical Institute at Olmütz from 18 to 21. He struggled to pay for his education, so he joined a St. Augustinian order to get support so he could continue his education. In this, he became Gregor, and he used that name from henceforth. He entered the Augustinian monastery of St. Thomas in 1843. This monastery was in the city of Brunn, and it's now Brno in the Czech Republic. He became a respected substitute teacher and passed numerous exams in his effort to become a biology professor. Unfortunately, he failed while taking the oral qualifying exam to become a professor in 1850, at 28 years old. So in 1851, Gregor Mendel went to the University of Vienna, where he remained until 1853. Vienna was the capital of the Austrian Empire, and it's currently the capital of Austria. There, he received education in the methods for performing experiments in mathematics and physics. He used this design and experimentation for the experiments that he did on the peas. Probability calculations that he found also helped make him a more exact meteorological researcher. Then, again, he went to earn his teaching credentials. In 1855, at the age of 33, he failed the oral portion of the exam again. Then he went back to the monastery. At the monastery, he embarked on the 8-year research project that would posthumously earn him his fame. In 1865, Mendel confronted current opinions of his results in 2 lectures. His findings were determined to be controversial due to them being different from the prevailing views at the time. Scientific Society members, however, felt the results were too unimportant to replicate, so they dropped him. He was the first one to use the principles of mathematics to describe biological phenomena. This approach to using science and reason in hybrid science was considered to be strange and unfamiliar to his audience. Only one researcher was actually known to have responded to Mendel's work. In 1868, Mendel became the abbot of the monastery. During the entirety of the time he was at the monastery, Mendel worked with bees, meteorology, and the plant Hericium, in addition to his peas. Mendel was the first one to ever make a scientific description of a tornado, which he did in 1871. In 1884, he died from kidney infection. Our modern understanding of how traits may be inherited through generations comes from the principles proposed by Gregor Mendel in 1865. Gregor Mendel spent 8 years doing experiments with pea plants, and based on his experiments, Mendel proposed 3 foundational principles of inheritance. Today, whether we talk about pea plants or human beings, genetic traits that follow the rules of inheritance that Mendel proposed are called Mendelian. So even today, we deal with Mendelian genetics. Mendel was curious about how traits were transferred from one generation to the next generation. Peas were a good model system because he could easily control their fertilization by hand pollinating them. Hand pollination involves transferring pollen with a small paintbrush from the anther to the stigma. This pollen could come from the same flower in what is known as self-fertilization, or the pollen could come from another plant's flowers in what is known as cross-fertilization. Before Gregor Mendel, the main idea for heredity was called blending. In this theory, you were a blend, or a mix, of both your parents. Like how yellow and blue paint mix to make green paint, people thought that the 2 parents' traits blended together to make some new combination of the traits. This idea was okay, but it had some obvious problems. some traits didn't actually seem to blend. Mendel's data did not support the ideas about trait blending that were popular among biologists of his time. There was never any merging of traits. There was never any greenish-yellow seeds or light purple flowers. Traits always looked like one of the two parents, but it wouldn't be a combination of both. So the offspring would make green or yellow seeds, but not greenish-yellow seeds. And they would make purple or white flowers, but not some weird mix of the two. Mendel concluded that blending was not the expected outcome of parental trait combinations. He hypothesized that each parent contributes some sort of inheritable particulate matter to the offspring. He called this heritable substance elementin. In 1865, Mendel did not know about DNA or genes. 1869 was when nucleic acids were first discovered. So what makes up DNA was first even discovered in 1869, 4 years after Mendel did his research. And DNA was first demonstrated to be heritable in 1944. So nearly 100 years later, we actually figured out about DNA. For each of the traits that Mendel examined, Mendel focused on how the elementin was distributed among the progeny. We know now that a single gene controls a single trait, and that elementin that he found is actually the assembly of those genes on chromosomes. Multiple forms of those genes, also known as alleles, represent different traits. For example, one allele could result in round seeds, while another allele would result in wrinkled seeds. When Mendel's results were rediscovered in 1900, the principles that he focused on were used to explain heredity and establish genetics as a science. Okay, let's get into Mendel's experiments. So this is some background. First, Mendel observed plant forms and their offspring for 2 years as they self-fertilized, also known as selfed, and ensured that their outward measurable characteristics remained constant in each generation. During this time period, Mendel observed 7 different characteristics in the pea plants, and each of these characteristics 2 main forms. Mendel looked at the 2 alternate forms of 7 different pea plant characteristics. The characteristics are seed color inside the seed, seed shape, seed coat color, pod shape, flower position, and stem length. The seeds could be either yellow or green in color, and they could have either a smooth or a wrinkled shape. The pea pods could be either yellow or green, and have either an inflated or a shriveled shape. The position of the flowers on the pea plant could be either axial or terminal. Axial flowers occur along the length of the stem, while terminal flowers occur at the tips of the stem. And then lastly, the height of the plant could be either short or tall. In the years Mendel spent letting his plants self, he verified the purity of his plants by confirming, for example, that tall plants only had tall children and tall grandchildren and so forth, because the 7 pea plant characteristics tracked by Mendel were consistent in generation after generation of self-fertilization. These parental lines of peas could be considered pure breeders or true breeders of the trait. In modern terminology, they were homozygous for the traits of interest. Mendel not only crossed pure breeding plants, he also crossed hybrid generations and crossed the hybrid progeny back to the parental lines. And these crosses, which in modern terminology, the ones we're going over, are F1 and F2, these are the classic crosses to generate genetically hybrid generations. Mendel designated the parents of the cross as P1 and P2, the first progeny as filial 1 or F1, and the second progeny as filial 2 or F2. So let's look at that. So parent 1 and parent 2 was how he designated them, and then filial 1, and then he crossed the filial 1 with itself to get filial 2. There are some definitions we should get into before we talk about Mendels experiments. So for the first one, it's gene. A gene is a heritable section of the chromosome that codes for a particular trait. An allele is a variation of a gene. Alleles are often expressed in uppercase or lowercase versions of a single letter. For example, 2 uppercase P's would be 2 alleles that express a homozygous dominant genotype. So, speaking of homozygous dominant, homozygous is when both alleles are the same, and then dominant means the allele would be expressed over the other allele. Recessive means the allele would not be expressed over the other allele. So when we draw it, it looks like this. So here we have 2 uppercase P's, and that is homozygous dominant. Then, if we have both alleles different from each other, it would be heterozygous. The recessive and the dominant allele are both there, and the dominant allele is expressed over the recessive allele. And then lastly, we have homozygous recessive. These are both the same, and they would both not be expressed if there was a dominant allele. So in this one, you only have the recessive trait. And this is the only option where the recessive trait is expressed. Heterozygous and homozygous dominant both have the dominant trait expressed. When we're talking about genes, we need to be considering both the genotype and the phenotype. The genotype is the code that's actually in the DNA, and how that genetic code codes for dominant and recessive alleles. Terms like homozygous dominant and heterozygous belong in this category. Phenotype is what the organism actually looks like. It's the observable trait that the organism has. Terms like purple petals or brown hair belong in this category. Phenotypes are both the genotype and the environment, so we need to take into account the genetic structure of an individual and any environmental factors that may have played a role. So an example of phenotype would be white flowered peas with a white flower color. The genotype for those white flowered pea plants would be 2 lowercase p's. Any letter could be used to represent these alleles, but typically the first letter of the dominant trait is used. So in this case, when we're talking about purple flowers, which are the dominant trait, and white flowers, which are the recessive trait, we use purple, the first letter of that, for the alleles. Before we get into the actual experiments, we should do some disclaimers. So, the ratios expressed throughout this presentation are just that. They're ratios. They provide the odds of an event happening, but they don't guarantee that that event will happen. So just because there is a 3-to-1 ratio of purple flowers to white flowers does not mean that if you plant 4 seeds, there will be 3 purple and 1 white seed. That's just the odds of the offspring falling into these categories each time. There is an independent chance for each of those seeds to be either of those phenotypes. So there's an independent chance that the offspring will fall into one of those 2 categories each time. And just because a trait is dominant does not mean that it will be more common in the population. So just because it is expressed over the recessive trait does not mean it's going to be common. For example, polydactyly is a disorder that results in having extra fingers or toes. Polydactyly is dominant over having 5 fingers on each hand. But we don't see many people with extra fingers or toes. And this is because even though the trait is dominant, it is not widespread in the population. So without further ado, let's get into Mendels Experiments. Mendels Experiments cross-pollinated one variety of purebred plant with another variety of purebred plant. And these crosses yielded offspring that looked like either one of the parent plants. So it looked like one of the parents, not both. It was not a blend of the 2 plants, like was previously thought. One of Mendel's monohybrid crosses was for different colored pea flowers. There were 2 different colors of pea flowers in his field, purple flowers and white flowers. In this case, the white plant only had white alleles, and the purple plant only had purple alleles. So he took a white flowered plant and a purple flowered plant, and he crossed them. When 2 true breeding plants are bred together, it's called hybridization. And the offspring of those parents is called the F1 generation. The F1 generation all came out purple. So let's get into that. So, there was the parents, 1 white and 1 purple. And we have the genotypes here. The white we found out was recessive because the offspring were all purple, and the purple was dominant. So we have lowercase and lowercase for the white, and we have uppercase and uppercase for the purple. And when they are crossed together, they make the F1 generation. And this is expressed in the Punnett square below. We figured out that the offspring, the F1 generation, were all heterozygous when we crossed and we had lowercase p from the white parent on this side And then we had uppercase P and uppercase P from the purple parent on this side. And then we took and we added them up. So the little p and the big P come together. And it goes the same across all the boxes. And if you notice, all of these boxes make the same uppercase and lowercase allele pattern. So all of them are heterozygous. So we figured out that the F1 generation is all heterozygous. So then, let's look at what he did next. Mendel took the F1 generation with its heterozygous genotype and he crossed it with itself. So this made the F2 generation. So I'm just going to draw this out. So we have the F2 generation, and that has 3 different allele frequencies. But it only has 2 different phenotype frequencies. So, the F1 generation, 2 hybrids, 2 heterozygous were crossed together, and we did this Punnett square. So uppercase P, lowercase p, uppercase P, lowercase p. And those 2 uppercase P's come together, and then they each come together for their square. So lowercase p, uppercase P, lowercase p, lowercase p. And we get these options. So for genotype, we get 1 that's homozygous dominant. We get 2 that are heterozygous. And we get 1 that is homozygous recessive. So for genotype, we get a 1 to 2 to 1 ratio. For phenotype, this is what color we end up getting. We get 3 that have at least 1 purple allele. So 3 of them are going to be purple. And then we have only 1 that ends up being white. So in the F2 generation, we have 3/4 of the plants that are purple and 1/4 of the plants that are white. The purple allele is dominant over the white allele. And this is a monohybrid cross, or a cross for a single trait. Monohybrid crosses always result in a 3-to-1 inheritance model. In another example, Mendel cross-pollinated plants with wrinkled seeds to those with round seeds. And he didn't get progeny with semi-wrinkly seeds like you would expect in blending. Instead, his offspring from the cross only had round seeds. So if we go through, Mendel took a round seed plant and a wrinkled plant and crossed them together, and the offspring were all round. All the offspring for this, the F1 generation, had 1 allele from the recessive trait, the wrinkly seeds, and 1 allele from the round seeds, the dominant trait. So all the offspring expressed round seeds as their phenotype, and they were all heterozygous. Because of this trait, Mendel proposed his first principle, the principle of uniformity. This principle states that all the progeny of a monohybrid cross will appear identical. So, whenever he did a monohybrid cross like this, with one dominant trait and one recessive trait, all of the offspring were identical, and they all expressed the dominant trait. So then, he took the F1 generation and crossed it with itself. And then the F2 generation, because of that cross, had 3 seeds that were round to every 1 seed that was wrinkled. Now for this one, we've got a genotypic ratio that has 1 homozygous dominant for every 2 heterozygous to every 1 homozygous recessive, just like the last time. So homozygous dominant, 2 dominant alleles, heterozygous, 1 that's each, and then homozygous recessive, 2 copies of the recessive allele. So we get this 3-to-1 proportion for phenotypes and a 1 to 2-to-1 proportion for genotypes. This 3-to-1 proportion from the F1 crosses suggested that there was a hidden recessive form of the trait. And Mendel recognized that this recessive trait was carried down to the F2 generation from the earlier parent generation. So whenever we do a Punnett square for a monohybrid cross, it's always going to result in a 3-to-1 ratio, with 3 of the dominant trait for every 1 of the recessive trait. And that's going to be in the F2 generation. The genotypic ratio for a monohybrid cross will always be 1 homozygous dominant to every 2 heterozygous for every 1 homozygous recessive. So a 1 to 2 to 1 ratio. This monohybrid cross can also be used to determine the genotype of an unknown but dominant trait expressing plant. So to figure that out, we cross the dominant trait expressing plant with a recessive plant. And if the unknown is homozygous dominant, none of the offspring will express the recessive trait. So like we did here, if unknown is homozygous dominant, it all results in heterozygous in the offspring. However, if the plant was heterozygous, then half the offspring will express the recessive trait, because half of them will only have the 2 recessive alleles. To summarize Mendel's experiments with pea plants for monohybrid crosses, first Mendel took the stigma and dusted it with pollen from another plant or with itself. The pollinated plant produced offspring, and that offspring was all heterozygous, and that offspring all expressed the dominant trait. Then Mendel self-fertilized the F1 generation to give rise to an F2 generation. The F2 generation had 3 dominant trait expressing plants for every 1 recessive trait expressing plant. The result of Mendel's experiments illustrates several features that are necessary for scientific data. Mendel used multiple trials to verify patterns in the data, and he also used a large sample size. For example, in the round and wrinkled cross, he used over 400 plants. That's a whole lot of pea plants. Mendels experiments also demonstrate that there is variation in the results of an experiment. So for one cross, he could get 3.2 to 1, and when he ran the experiment again, he could get 2.9 to 1. Both of those crosses would round to 3 to 1, but due to chance, there was variation in the results he would get. Mendel's expression of notation for genes using a capital and a lowercase letter for the hybrid genotype actually represented what we now know as the two genes of one allele. Damn it. Start again. Okay. Mendel's notation of genes using a capital and lowercase letter, uppercase A and lowercase a, for example, actually represented what we now know as the two alleles of one gene. When one parent carried all the dominant traits, capital A and capital A, the F1 hybrids were indistinguishable from that parent. So, if there was one parent that was dominant, the F1 plants all had the same phenotype. But they had a different genotype. And that genotype carried the potential to look like the recessive plant. After observing this potential to express a trait without showing the phenotype, Mendel put forth his second principle of inheritance. And this principle of inheritance is known as the principle of segregation. According to this principle, the particles, or alleles as we now know them, that determine the traits are separated into gametes during meiosis, and meiosis produces equal numbers of egg and sperm cells that contain each allele. Mendel went into dihybrid crosses. So after he looked at single traits, he decided to look at two different traits. So he looked at crossing plants for two different traits. One of his experiments looked at a cross of plants with round yellow seeds and wrinkled green seeds. Oops. And this is shown here. So, we got round yellow and wrinkled green. The round yellow plant is homozygous dominant for both traits, and the wrinkled green plant is homozygous recessive for both traits. Those parents are crossed to make an F1 generation, and that F1 generation is completely round and yellow. So the traits that are expressed in the F1 generation are only those that are dominant. And to do a dihybrid cross, we have to take and foil these alleles. So we have the round one and the yellow one, and they both have dominant alleles. So we take and foil it, so we take the first two, and we get an RY, and then we take the outer two, and we get a capital RY. We take the inner two, capital RY, and we take the last two, and we get a capital RY. So foil, first, outer, inner, last. And you use it whenever you're doing a dihybrid cross. It's also a term that can be done for algebra. So any sort of simple algebraic equation uses FOIL too. Over here, we have foiling for the one that's all recessive. So first, we get the little r, little y, outer, little r, little y, and we put those here, inner, little r, little y, and then last, little r, little y. And then we do this whole Punnett square. Now, because all of these alleles are the same, these are all capital R, capital Y, and these are all little r, little y. I just drew in the first couple results of that. So when we take it and we put it through the Punnett square, we have capital R from this one and a lowercase r from this one. And then we have a capital Y from this one and a lowercase y from this one. We want to group all the alleles together for this genotype of the final plant. So the genotype for this plant is capital R, lowercase r, capital Y, lowercase y. So the F1 generation is heterozygous for both the round trait and the yellow trait. So then the next part is to take two F1 plants and cross them. So we have the F1 generation. When the F1 generation was self-fertilized, it resulted in the F2 generation. So let's look at the F2 generation. We've got all these round yellow plants, and they had this genotype. So when we FOIL that genotype, we take the first letter, so F, capital R, capital Y, the outer letters, capital R, lowercase y, so capital R, capital Y here, capital R, lowercase y there, and then the inner letters, lowercase r, uppercase y, there, and then the last two letters, lowercase r, lowercase y. Now, because they both have the same genotype, we can put it along the top of this Punnett square and along the side of the Punnett square. And then we take and we fill in the rest of the Punnett square with these. So this one has capital R, this one has capital R, so we combine the two. This one has capital Y, this one has capital Y. We combine those two. And we do the same for every pair of gametes that is across the Punnett square. And when we're finished, we look and see what the observed phenotypes are going to be. And if we look at it, there are 9 that have capital R and a capital Y at least. So at least one of each. So there's going to be 9 that express both dominant traits. So we're going to have 9 that are round and yellow. And then there are 3 that express the capital R, so they're going to be round, but they don't have any capital Y. So those are going to be round, and they're going to be green. So we have 3 that are round and green. And then we also have 3 that are going to be yellow, because they have the capital Y, but they don't have the capital R. So they're going to be wrinkled. So we have 3 that are yellow and wrinkled. And then lastly, we have 1 that expresses both of the recessive traits. So we have 1 that is green and wrinkled. So whenever we do a dihybrid cross, we get 9 of the individuals expressing both dominant traits to every 3 of the individuals expressing one of the dominant traits, to every 3 of the individuals expressing the other of the dominant traits, to every 1 of the individuals that express both of the recessive traits. So, based on the concept of segregation, Mendel predicted that traits must sort into gametes separately. And by extrapolating from his earlier data, Mendel also predicted that the inheritance of one trait did not affect the inheritance of the other trait. So when we looked at this, the inheritance of yellow did not affect the inheritance of the seed shape. So seed color did not affect the inheritance of the seed shape. Mendel tested this idea of trait independence with more complex crosses as well. So he took plants that were purebred for one dominant trait and one recessive trait, and then crossed them together. And as expected, the traits were all observed to be dominant in the F1 generation, regardless of what plant they were from. So even though the recessive traits were not grouped together and the dominant traits were not grouped together, the F1 generation still only expressed those dominant traits. The traits behaved as though they were segregated independently. From this data, Mendel developed the third principle of inheritance. This is the principle of independent assortment. According to independent assortment, all alleles at one locus segregate into gametes independently of all the alleles at another locus. So a locus is just a spot in the DNA where the gene is. So the alleles at one spot will separate independently of alleles at another spot. And the gametes will have equal opportunity to have one allele or the other allele. Mendel came up with 5 rules of inheritance. Okay? So the first one is this: genes exist. And that there are 2 different versions of a gene that can be inherited. Those different versions of a gene are called alleles. The second thing is that one version of a gene is inherited from each parent. So one parent donates one allele. Third, one allele is dominant or expressed over the recessive allele. So one allele is expressed over the other allele. 4, the law of segregation. Alleles separate from each other during gamete formation, and then they end up in different gametes. Each gamete or sperm, egg or sperm, ends up with just the one allele. The second allele is inherited during fertilization of the gamete. In all basics, gametes are haploid, developing organisms are diploid. And then 5, the law of independent assortment. So these say that the traits inherit or separate separately from each other independently. So each allele has an independent trait, independent chance to be inherited. The law of independent assortment is an ideal law, but it does not always apply. Independent assortment is a good factor for looking at crosses with more than one trait. So when we did the Dihybrid Cross, we needed to take into account that both of those genes, the gene for round seeds and the gene for yellow seeds, each inherited independently of the other. More lasting than the P data that Mendel presented in 1862 has been his methodological hypothesis testing and careful application of mathematical models to study biological inheritance. He was one of the first to really use mathematics to study biological phenomena. From his first experiments with monohybrid crosses, Mendel formed statistical predictions about trait inheritance, and then he tested those traits with more complex experiments on dihybrid and even trihybrid crosses. This method of developing statistical expectations about inheritance and then testing them is one of the most significant contributions that Mendel made to biology. Even though many organisms do show inheritance patterns similar to the ones that Mendel described, the main important things that Mendel had impact with in science are his principles of inheritance. So his principles of inheritance had much greater impact than his original pea plant manipulations. And to this day, scientists use Mendel's principles of inheritance to explain the most basic phenomena of inheritance. We can apply the ideas that Mendel had to pedigrees. Pedigrees are records of individuals in a family and how they express a specific trait. So let's look at this pedigree for widow's peak. Each of these generations, first generation, the second generation, and third generation, represents a horizontal row in the diagram. Square and circle symbols represent the male and females, respectively. Open symbols represent unaffected members, and then shaded represent members that have the affected trait. So in this case, shaded individuals have a widow's peak. A horizontal line connects the two individuals in a mating pair. So this male and female had offspring, and then the vertical line down connects the mating pair to the offspring. So vertical line down, horizontal line across shows all the offspring. And then those offspring had their mates, and they had offspring. So once we have the offspring, we have the numbers assigned to each individual, and we have the Roman numerals that represent the generation. So here we've got the Roman numerals for each generation, 1, 2, and 3. And then because there's so many individuals in this one, they are labeled with the numbers 1 through 15. So now that we've got what a pedigree is and how to make one, let's look at what this one tells us. So pedigrees show how traits are passed down in families in different patterns, and then they illustrate these patterns. So in this one, there's an affected female. So this female has a widow's peak, and then this male who doesn't have a widow's peak. And then those two had 4 children. The first one was a girl, the second one was a boy, the third one was a boy, and the fourth one was a girl. Those are individuals 3, 5, 7, and 8 respectively. 3 of those children ended up having children of their own. 1 of them did not, number 7. The children that are from 3 and 4 each had, they had 2 boys and a girl. 5 and 6 had a girl, and then 8 and 9 had 2 boys. So, of those, let's look at the ones that have widow's peaks. We know that we know that widow's peak is a dominant trait. So widow's peak is a capital W. Not a widow's peak is a little w. So this one has to have at least 1 capital W. And we don't know what that is. This one has to have 2 little w's. Okay? Because there's no widow's peak there. This one. Okay. So individual number 3 has a widow's peak. So at least 1 capital W. And her husband also has a widow's peak. So capital W. And then their children. This one does not have a widow's peak. So 2 little w's. This one has a widow's peak. And then I'm just adding a space there so that we know that there's another one that's needed. And then this one has a widow's peak. Now we can't figure out what these two are because we don't have any more data, but we can figure out what 3 and 4 are. So because they have one individual that doesn't have a widow's peak, they have to have 2 little w alleles. So we know that those 2 little w alleles have to come from both parents. So these two's genotype is going to be big W, little w. Over here, it's a similar thing. This one also doesn't have a widow's peak. And then she doesn't have a widow's peak. And their kid doesn't have a widow's peak. So they all have to have 2 little w's. And because this one has 2 little w's, we know that the mom has to have a little w. So her genotype is big W, little w. This one, we don't know what his genotype is going to be because he didn't have kids. So we don't have anything to tell. But for this one, we know that this one had to be at least 1 widow's peak allele, and she married someone who did not have a widow's peak. So, looking at the kids, the one kid does not have a widow's peak, so 2 little w's, and the one does, so 1 big W, and we don't know what the other one is. So, figuring that out, this one has to be a little w, because they had 1 kid who didn't have a widow's peak. So, that's how we can figure out the specific patterns of inheritance through a family. Dominant traits are more expressed through a family over generations. And pedigrees show the history of a specific trait through a family. So, Mendelian genetics is an important concept, and it provides an ideal version of how genetics work. But, and in nature there's always a but, unfortunately, there are always exceptions. Mendelian genetics assumes 3 main things. It assumes that there is 1 gene per trait, and that there are 2 alleles or options for every gene, and also that there is a clear dominant and recessive relationship between the alleles. So, it assumes there's 1 spot in the DNA that we're looking at, there are 2 options at that spot, and that one of those options is expressed over the other option. Most genes don't follow one or more of these assumptions. For example, none of my microsatellites follow any of these. So Mendelian genetics does not work for my microsatellites. There are things like incomplete dominance, multiple alleles, and genes that correspond to multiple phenotypes that all just don't follow the Mendelian genetic patterns. So let's get into the different genetic models that don't follow Mendel's inheritance patterns. The first of these models is sex-linked genes. Sex-linked genes typically follow Mendelian inheritance rules, but they have a twist. Because sex-linked genes are located on the X chromosome, females have 2 copies, and males have 1 copy. So if there is a recessive trait on the X chromosome that a male has, it will be expressed. So, if a recessive allele for colorblindness, because colorblindness is a recessive disorder, so if there's a recessive allele there, females would be able to mask that allele with a functional copy, following the normal rules of Mendelian genetics. But males only have the 1 copy. They have that Y that blocks that they don't have another copy of the X chromosome. So they wouldn't be able to mask any recessive allele that they have. So in this example of sex-linked inheritance, with a color blindness example, we are looking at a recessive disorder. So normal vision is the dominant allele, and then recessive color blindness is the recessive allele. So we have this normal color vision male with the X chromosome, and then we write it in superscript here for the normal color vision, and then the Y for the male. And then the normal color vision female, that is a carrier for the color blindness allele. So she's got one normal allele and one recessive color blindness allele. But because Mendelian genetics is still in play, that dominant allele is going to be expressed. So when we cross these two, the normal colored male takes his allele, and then the female, her allele, and they make a normal vision female. They do the same here, and they get a normal vision female. This one is a heterozygous female, so she is a carrier for color blindness. And then over here, we have X and Y, so we have a normal vision male. And then over here, another male, but this one has the recessive allele. So this one only has the one allele and is recessive for color blindness. So this one is the one that's going to be affected. So because males only have the one copy, it's much more common to see males have a sex-linked disorder. Even though females can have sex-linked disorders, it happens at a much lower frequency. So let's get into the next concept that violates Mendelian inheritance. This is called incomplete dominance. Incomplete dominance is a violation of Mendelian genetics in a classic way. In incomplete dominance, heterozygous individuals have a combination of the two phenotypes. So instead of red and white having red and white, they would make pink. So it's sort of like the blending theory. And part of why that theory still, why it gained ground. Because in some plants, it does exist. So for example, in snapdragons, they have red flowers and white flowers. And when a red-flowered individual with two capital R alleles and a white-flowered individual with two lowercase r alleles are crossed, those two plants make pink flowers. So the heterozygous individuals for that are pink. So once we have that F1 generation that's all pink, a combination of those two alleles, we go to the F2 generation, and it results in a 1 to 2 to 1 ratio of red to pink to white, because it follows the genotypic ratios. So when we end up having the F2 generation, we've got one red flower for every two pink flowers to every one white flower. Another related violation of Mendelian genetics is codominance. In codominance, both the alleles are expressed in the phenotype. An example of codominance would be in cows. When a red-haired cow and a white-haired cow are crossed, the offspring typically have red and white spots. So codominance is when both the alleles are expressed. Incomplete dominance is when it looks like there's a bit of mixing. So incomplete, red and white make pink. Codominance, red and white make spots of red and white. There's also something called multiple alleles. So when there's more than two options. Blood type is an example of both codominance and multiple alleles. There are 3 different alleles for blood type. They are typically called A, B, and O. Both type A and B are dominant over O, and A and B are codominant. So when you have both of them on the surface of the red blood cells, you have type AB blood, because they're both expressed. The alleles are typically written for Punnett squares using the notation little I with the A, B, or O as a superscript. So for example here, little I and a big A, and little O, or little I and a little O. So A and B are capitalized, O is lowercase. A and B encode carbohydrates on the surface of the cell. A encodes galactosamine, and B encodes galactose. Type O blood has no carbohydrates on the surface of its cells. Additionally, type A and B make antibodies against whichever is not present. Well, all blood types make antibodies against whichever type is not present. So if you have type A blood, you would have anti-B antibodies. Of course, since O has nothing can really make antibodies against O. So if you have type A blood, you've got anti-B antibodies. If you have type B blood, you have anti-B antibody, or anti-A antibodies. So type A blood can't receive blood from a type B donor because of this. So when we're talking about blood type, we've got this entire table that we can make up about what type of blood that can be donated or received based on the carbohydrates. So based on the blood type A, B, and O. So if we have type A blood, it can receive A and O because A has the same type of as itself and O has nothing. And it can donate blood to type A and AB because both of those don't have antibodies against A. Type B blood can receive B and O because O has nothing. And it can donate to B and AB. Again, because both of those don't have B antibodies. Anti-B antibodies. And then AB can donate, or it can receive blood from all, because it can receive blood from A, it can receive blood from B, and it can receive blood from O, because it doesn't have antibodies against any of them. And it can only donate to itself, because all of the rest of these have some sort of antibodies against A or B. And then O. O can receive blood from type O, because nothing and nothing. And it can donate to all. So, because it has nothing on the surface of its cells, it can go to whatever, and there's no antibodies against it. But it does have antibodies against A and B, so it can't receive any of the other blood types. And that's sort of how that works out. If you have multiple alleles, things can get kind of complicated to explain. Next up is pleiotropy. Pleiotropy is when one gene causes multiple phenotypes. Most examples of pleiotropy are when an allele of a gene causes multiple symptoms of a disease. Genetic diseases are not the only example of pleiotropy. Skin color is an example of pleiotropy as well. For example, there's an MC1R gene, and this gene affects both skin color and hair color and freckles. So it can do a lot of different things. Skin color is also an example of polygenic inheritance. Polygenic inheritance is when multiple genes result in one main phenotype. So there's an accumulation of multiple genes together that all add up to the one phenotype. In this case, skin color is a big combination of a bunch of different genes coming together to make the skin color. Another example of polygenic inheritance is height. Multiple genes accumulate to produce one height. Epistasis. That is when the expression of a gene at one locus or one location alters the expression of a gene at another locus. So, in one thing, one gene decides whether the other gene will be phenotypically expressed. So, epistasis, when one gene decides if the other one is expressed. For example, in mice, there is one gene for pigment and one gene for no pigment. So this is a dominant and recessive relationship following simple Mendelian genetics. So if there is pigment, it's dominant. If there is not pigment, it is recessive. So if all the alleles don't code for any pigment, then the mouse will be white. It doesn't matter what color the pigment might be. If the mouse can't make any pigment, then there will be no pigment. So when the mouse can make pigment, then the genes that code for what color that pigment might be come into play. So in this pattern of inheritance, we get a 9:3:4 ratio, with 9 of the dominant pigmented mice, 3 of the recessively pigmented mice, and 4 of the non-pigmented mice. So let's look at that in a Punnett square. So here we've got pigment. So pigment is going to be a capital P, no pigment is going to be a lowercase p. And then under that, we've got the gene for the type of pigment. So here we have black, which is the capital B, and brown for the lowercase b, because black pigment is dominant over the brown pigment. So when we put it in a Punnett square, we have all of this, and then this one, all of these that are black. So we have 9 of the black pigmented mice. Then we have 3 of the brown pigmented mice. And then if they code for the gene that has no pigment, then it doesn't matter if that would code for black pigment or brown pigment. They still can't make pigment. So all of those would be white. And that's how we get that 9:3:4 ratio. Another thing to take into consideration for phenotype is the environment. Genes are not the only thing that can cause a phenotype, because phenotypes are the result of a genotype and the environment together. For example, a hydrangea can change color based on the pH of the soil that it's in. So in one pH it could be blue, but in another pH it would be pink. And a pretty common form of environment consideration, people can use a compound to alter their hair color. So people dye their hair a color that's different from what they would normally express as their genotype. In Siamese cats, the extremities of the cat get dark as the temperature goes down to keep them warm at night. So their hair is dependent on the environment, temperature. Gene expression dictates the phenotype, and then the environment affects the phenotype. In humans, because we need to take into consideration the fact that experiments are typically frowned upon, we usually use pedigree trees to determine how traits are inherited. Recessive traits skip generations because of heterozygous carriers. The carrier has one normal copy of the gene and one affected copy of the gene, so they only express the normal copy. It takes two carriers, or a carrier and an affected individual, to make affected offspring. Thus, there is typically a low prevalence of recessive traits in the population. Recessive traits being expressed anyway. However, there is an exception. In consanguineous marriages, also known as inbreeding or endogamy, there is an increase in the odds of two carriers coming together. This increase in problems for consanguineous marriages is why those marriages are not done. Because where there's an increase in the recessive genes that are expressed in that population. And recessive genes are more commonly the ones that are problematic. So, some dominant genetic conditions are things like dwarfism or Huntington's disease. Dominant genetic conditions are generally prevalent in the population generally exist in the population. They normally aren't prevalent, but they generally exist if they're not fatal or if they manifest later in life. Dwarfism is an example of a condition that is not fatal, and so that continues to be present in the population. Huntington's disease is a genetic disease that's stuck around because the disease usually manifests after the children are already born, and then the disease comes into play. But recessive and dominant traits, they don't have to be disease specific. They can be many different things. So things like earlobes are free or attached, where attached earlobes are recessive and free earlobes are dominant. Albinism is also a recessive trait, and that is when you don't make enough pigment. So that is also in the population. There are lots of different traits that just are around, and Mendelian genetics explains a good portion of them, and non-Mendelian genetics explains the rest. So Mendelian genetics helps to determine the phenotypes that an offspring will have and how heritable traits are passed through generations. Current research still includes Mendelian genetics. In 2013, research by Lin Li and many others looked at maize shoots, so corn root shoots, and they did RNA sequencing on them for 28,603 genes. They determined that 145 of those genes showed inheritance patterns that were reflective of Mendelian genetics, and 210 of the genes were not reflective of Mendelian genetics. From what I've read in the paper, it seems that they were unable to determine the inheritance pattern for the rest of the genes. Thank you for watching this video. Again, my name is Janae Heiss. I hope you learned something about Mendelian genetics, and I'll see you in the next video.